Hey guys, I wanted to pick up where we left off in class to make sure we got through everything. And let's take a look at what you should have seen in the microscope. This was just supposed to be the fresh water, okay? And you guys probably noticed that the Allodia, they have a really kind of regular shape to their cells, and that's because they have a cell wall that's basically the shape of a box. And in the microscope, at this point, you can't really see the cell membrane at all. Because it's pretty much flush up against the cell wall. All right, so if you can imagine, here's the cell wall, the cell membrane or the plasma membrane is right up against it. And then you also see all these chloroplasts, and they're kind of everywhere. Now, when we switched it to salt water, now you guys probably saw that each cell really had just kind of this ball of chloroplasts, and maybe you couldn't even tell the individual chloroplasts. It kind of just looked like one big clump of them. And so what is actually going on here, so the cell used to be this big, and now the cell is only this big, okay? And so all the chloroplasts have squished together. And from here to here, what has happened is water has come out. Okay. And this happens to our cell in the presence of anything that would dissolve in water. So things like salt and sugar. So you guys may have noticed if you drink a really sugary drink, like, I don't know, Body Armor, it's probably going to dehydrate your cells if it's concentrated enough, and especially things like soda, right? So you're surrounding your cells with sugar, and that draws out water. So let me show you what this looks like on a molecular level. Where am I? Okay. Hang on, I'm going to refresh this. All right, so normally, as we talked about in class, water is able to permeate the cell membrane. So you just notice the molecules are just kind of freely moving back and forth, no net movement either way. Now, if we add salt, okay, so let's see what happens. So they're still moving back and forth, but there is a greater number moving in this direction to this side, which is why the water level rises. And that is due to the charged nature of this solute. Okay, anything that dissolves in water, we call it a solute. And so this is positively charged, so it's going to hydrogen bond with those oxygens, and this is negatively charged, so it's going to hydrogen bond with the hydrogens, and it's going to basically attract over more water. Okay, so you can see some water is still able to go back and forth, but a lot of them are attracted to the salt. And so that's really what you did. Let's go back to our picture here. Let me get this going. All right, hang on. It's quite a process here. All right. There we go. Okay, so anytime you put salt, okay, so imagine these are our salt crystals, around a cell, that's going to attract water out. Okay, whereas here there's no salt, so any water that leaves will probably get replaced by some more water that's outside of the cell. So there's an equilibrium of water here. Here, though, the water is being unequally drawn out and attracted to these solutes. Okay, so this picture is a good one because it explains the terminology that you guys are going to use to explain this phenomenon. All right, this is normal, okay? Okay, this is isotonic, which means the number of solutes outside of the cell is the same as inside the cell. Okay, so that means there's some solutes on the outside, some on the inside. There's sort of this equal exchange going on here. Whatever goes in must come out, okay? This is not a great situation for plants. We don't normally feed plants water with solutes in it. Okay, so what will happen in this case, the plant won't look real perky. It's going to be what we call flaccid. This is going to be like when your plant looks a little droopy, right? Okay, that would be what its cells would look like. Now, the Allodia that we looked at first was turgid. So this is a plant that looks very perky like this. Okay, because all of its cells are getting fresh water. Okay, so this is fresh water. Okay, so 0 molar. okay, here. The inside of the cell is somewhere around 0.2 molar here, or 0.1. Let's say 0.1 here. So there's more solutes inside, so more water is going to enter. Okay, I want you guys to write down water follows solute. Wherever the most solutes are, that's the first thing you have to figure out. That's where the water will go. And so water in this case would enter the cells and put pressure on the cell wall, and that's really what's going to hold the plant up. All right, so the cell's going to grow. Now, the solution we say is hypotonic. Hypotonic means it has less solute. Okay, so fresh water has less solutes than the inside of the cell. So we can say the solution is hypotonic. We can also say, though, that the inside of the cell is hypertonic. Hypertonic means more solute. So whenever you use these terms, you want to make sure you say where you're talking about. So this is hypotonic solution. Okay, or you can say, remember, hypertonic cell. Okay, so now if we place the cell into, like, say, what we did with the salt water, okay, let's say we put it in a really salty solution, like 0.8 molar, okay? We said the inside of the cell is usually 0.1 molar. Okay, way more solutes out here, way hypertonic out here, very hypotonic in here. And so since that's where the solutes are, you imagine there's lots of salt molecules here, they're going to draw the water out, all right? And remember we said the cell membrane shrinks back, and that's plasmolysis, okay? The cell has plasmolized, or it's undergoing plasmolysis. Either way. Okay, so this is plant cells like we saw today. Now let's look at an animal cell. So animal cells are a little different. They don't have the cell walls, so different things are going to happen. Okay, let's look at the isotonic condition. So that means there's some solutes on the outside, but the same concentration of solutes on the inside. Okay, so everything's equal. So water coming in equals water going out. Okay, so we just say this is a normal cell here. Okay, now hypotonic here. So this means that we're putting it in water that doesn't have any solutes. So this would be like if we put it in fresh water. Okay, notice there's a bigger arrow going in. Okay, so whatever small amount of solutes inside the cell, they're going to attract this fresh water. So you notice the cell blows up, and actually it'll end up bursting. Okay, this is called lysis. All right, or cell lysing just means that it breaks open. So if a red blood cell was placed in fresh water, they would all burst because they would fill so far with water. Okay, what if we put a blood cell in hypertonic solution? So that means really, really, really concentrated. Okay, this is like if we put it in salt water. All right, so now you notice big arrow out because water follows the solutes. So this thing is really going to shrink back. And we can call this plasmolysis as well. Okay, so lysis only happens in animal cells because they don't have the cell wall to protect it from that. Okay, so you got to remember that. All right, now we are going to wait. Next class, we're going to talk about this thing called water potential. So in general biology, we learned about osmosis. We learned kind of what happens, which way the water goes. But now that you guys are in AP Bio, we're actually going to quantify osmosis. So we're going to talk about this stuff, okay? And we'll practice more about that. All right? So I'll see you guys next week.